Hi Genetic Innovation students, and welcome to your next lecture in Section 2 on recombinant DNA technology. In this lecture, we'll be moving on to the next learning objective, which is to illustrate the process of molecular cloning and to describe how and why the technique is used. Molecular cloning is a technique that's used to create multiple copies of a DNA fragment or a protein. And the ability to replicate a gene or a protein is a fundamental tool in molecular biology because we need to create multiple copies of a gene or protein in order to study its structure. In addition to basic science, molecular cloning also has other uses in applications such as creating genetically modified organisms, such as pest-resistant crops. Molecular cloning is also used to create bacterial biofilms that have the ability to clear toxic waste. Cloning of a protein or creating multiple copies of a protein is also important in the preparation of monoclonal antibodies, which are used both in research as well as in drug therapies. The basic workflow of molecular cloning involves 4 major steps. Firstly, we need to isolate the target DNA fragments. We then need to ligate that target DNA fragment into a cloning vector in order to create a recombinant molecule. That recombinant DNA fragment is transformed into a bacterial host or host organism in order for it to be propagated. Transformed bacteria are then screened to select for the host that contain the intended recombinant DNA plasmid. We'll now have a look at this diagram which summarizes the workflow of molecular cloning. We'll begin with a plasmid DNA fragment that can replicate independently in a bacterial host. That plasmid DNA fragment is isolated. The gene of interest from the target cell is also isolated. The next step is to create a recombinant DNA fragment or plasmid. This recombinant fragment is made up of the plasmid that can replicate in the bacterial host with the gene of interest inserted into that fragment. The gene of interest, or the DNA fragment that we wish to study in further detail, is referred to as the insert DNA fragment. The plasmid containing the insert DNA fragment is then inserted into a bacterial host, and this is now called a recombinant bacterium. The recombinant bacteria are then allowed to replicate themselves or propagate in culture overnight. During propagation, multiple copies of the bacterium are created, and in addition to the bacterium replicating itself, the inserted plasmid DNA fragment also replicates within that bacterium, thus creating multiple copies or millions of copies of the insert DNA fragment. Once that's replicated, we now have many copies of our insert DNA fragment. DNA can then be isolated from the bacteria, and that insert DNA fragment can be isolated to be studied further in basic research. Alternatively, the gene of interest can be inserted into a plasmid that allows for the expression of that gene. That gene is then expressed by the bacterium, and multiple copies of the protein are then produced inside the bacterium. And the protein can then be harvested and also used in basic research on the protein structure. Now that we've covered the overview of how molecular cloning works, we'll now be going through each of these steps in detail. In previous sections, we've already covered restriction enzymes, which are used to cleave DNA sequences. Now that we know how DNA sequences can be cut or cleaved, we'll now discuss how to rejoin these DNA fragments. A DNA ligase is an enzyme that can catalyze the formation of a phosphodiester bond between the 5' phosphate and 3' hydroxyl group of 2 separate DNA sequences. DNA ligases originally evolved in order to repair single-stranded DNA nicks in DNA fragments. These ligases are able to join both blunt and cohesive ends. This diagram shows how a DNA ligase can be used to join two separate fragments of DNA. DNA fragments from two separate sources are both restricted with the same restriction enzyme, EcoR1. As they are restricted with the same enzyme, which detects a specific target sequence, they now have complementary overhangs. These complementary overhangs can align with each other. However, the covalent bond or phosphodiester bond can only be created when a DNA ligase is introduced into the reaction. The DNA ligase can then repair what is recognized as nicks in the backbone and seals the two fragments, thus covalently bonding the two strands of DNA. T4DNA ligase is an ATP-dependent enzyme that was isolated and cloned from the T4 bacteriophage. It's now a very widely used tool in molecular biology that we commonly use in DNA ligation reactions. Now let's discuss a little about plasmids and vectors. A plasmid is an extrachromosomal double-stranded DNA molecule which is usually circular. A plasmid has the property of replicating independently from the host chromosome within a bacterial cell. A vector is a form of a plasmid that is used as a carrier DNA molecule. The vector has the property of replicating a cloned DNA fragment in the host cell. Vectors are designed to replicate independently, and they also have several restriction enzyme sites that allow for DNA fragments to be inserted into these vectors. Vectors also carry selectable marker genes. These selectable marker genes allow the researcher to distinguish between host cells that have taken up the vector and those that have not. Plasmids are often used in DNA cloning. Genetically modified bacterial plasmids were the first vectors that were developed. All plasmids must contain an origin of replication in order to allow for them to replicate within the bacterial host. Plasmids are also engineered to contain a multiple cloning site. The multiple cloning site is a region of the plasmid that contains the restriction enzyme sequences or target sequences of commonly used restriction enzymes. In addition to the multiple cloning site, we also have a marker gene. An example of a marker gene is an ampicillin resistance gene or an antibiotic resistance gene. And this marker gene allows for the selection of host cells that contain the inserted vector. Plasmids may also contain DNA sequencing primer sites in order for these plasmids to be used in DNA sequencing. Although it's not shown in this diagram, plasmids may also contain a promoter upstream of the multiple cloning site. This is a common feature of expression vectors that are used to express proteins. We will now discuss how a DNA fragment can be inserted into a vector. DNA fragments of interest, or insert DNA fragments, are ligated into a vector in order to create a recombinant plasmid. And there are various methods in which this can be achieved. The first method that we'll look at is non-directional cloning. In non-directional cloning, the insert DNA fragment is inserted into the vector in any orientation. So if you look at this insert fragment, for example, which has a 5' GATC overhang, the 5' GATC overhang is present on either side of the insert DNA fragment. So if this insert fragment were to flip around, it would still be able to be ligated into the vector. And this is an example of non-directional cloning. There are various methods of non-directional cloning. These include TA cloning, blunt end cloning, and single restriction digests. Non-directional cloning is used for gene studies where the orientation of the insert DNA fragment does not affect the outcome of the experiment. The second method of cloning a DNA fragment into a vector is directional cloning. Directional cloning involves a double restriction digest where we use two different restriction enzymes. So if you look at this diagram, you'll see that the insert DNA fragment can be ligated into the vector in one direction only, and that's why this technique is called directional cloning. TA cloning is the simplest and most efficient form of non-directional cloning. TA cloning is based on the property of Taq DNA polymerase. PCR products that are amplified with Taq DNA polymerase leave 3' overhangs. This property is used to our advantage for TA cloning. Cloning vectors are engineered to contain 5' T overhangs, and this allows for the 3' A overhang and the 5' T overhang to ligate to each other. This cohesive overhang promotes binding of the vector and insert DNA and also prevents religation of the vector to itself. Another method of non-directional cloning is blunt end cloning. Blunt end cloning is used for various reasons. For example, if we're unable to find an appropriate restriction enzyme site in the insert DNA fragment, we can use a high-fidelity enzyme that doesn't leave overhangs. Alternatively, any overhangs on the insert DNA fragment can be cleaved with an exonuclease in order to produce blunt ends on the insert DNA fragment. For example, a clenofragment can be used to remove any 3' overhangs on the insert DNA fragment and also adds bases to blunt 5' overhangs. A restriction enzyme such as EcoR5 can be used to cleave the vector. EcoR5 cleaves in the center of its restriction site, and so it does not leave any overhangs. So now we have an insert which doesn't have overhangs, as well as a vector that has no overhangs. And we want to ligate this insert DNA fragment into the vector. Due to the fact that there are no overhangs in this technique, it is less efficient than TA cloning. However, the hydrogen bonds formed between the 5' phosphate and the 3' hydroxyl are still sufficient to facilitate an interaction between vector and insert DNA and allows for the DNA ligase to ligate the two fragments to each other. So this slide summarizes the two non-directional cloning techniques that we just discussed, which is TA cloning and blunt end cloning. Now, say for example, we have an insert DNA fragment with A overhangs, but we only have a blunt end cloning vector. Or we have a TA cloning vector with T overhangs, but we have an insert DNA fragment without any overhangs due to the fact that it was amplified from a high fidelity polymerase. You can add a DA tail to the insert DNA fragment in order for it to have the A overhangs so that it can be inserted into a TA cloning vector. Alternatively, you could blunt the ends of an insert fragment that was amplified with Taq DNA polymerase and remove the A overhangs so that it may be inserted into a blunt end cloning vector. So this means that there's quite a lot of variability in the way that these techniques can be conducted, and we can adapt our experiment to any situation. The final technique that we'll discuss in non-directional cloning is a single restriction digest. In this procedure, one restriction enzyme is used to cut both the vector and the insert DNA fragments. This results in the formation of 5' or 3' overhangs in both the vector and insert DNA. Hydrogen bonding between the cohesive overhangs facilitates vector and insert DNA interaction, and the fragments can then be ligated. As I mentioned before, this is a non-directional method of cloning because this insert DNA fragment can go in any orientation. If you were to draw this insert DNA fragment in your notebook, you would see that it could go in any orientation. If you were to flip the whole sequence around, it can go into the vector in any orientation. The single restriction digest is much more efficient than blunt end ligation, and it's the most commonly used technique in non-directional cloning because it also relies on fewer restriction enzymes, and there's fewer steps involved in this protocol. Now we'll move on to directional cloning. Directional cloning is used for gene expression studies. This involves the double digestion of both the vector and the insert DNA fragment. And this technique is done to ensure that the insert DNA ligates into the plasmid in the correct orientation. Here we have an example of the insert DNA fragment and the vector. Both the insert and vector DNA fragments have been restricted with HinD3 and KPN1. And so now we have cohesive overhangs on both sides. So there's a different overhang on either side. We have the HinD3 restriction enzyme site on the left and the KPN restriction enzyme site on the right. And the vector has also been restricted with the same two restriction enzymes. The insert DNA fragment can only be ligated into the vector in this direction. If you were to do a 180 degree rotation of this insert DNA fragment, you will see that it can no longer fit into the vector. And so it can only go in this linear orientation. In this way, directional cloning is achieved. So if we have a DNA sequence that needs to be expressed, we can ensure that the coding strand and the template strand are on the correct sides of the cloning vector. Now we'll discuss another way directional cloning can be achieved. So say, for example, we have a vector and it contains certain restriction enzymes in its multiple cloning site. And these restriction enzymes that are present in the multiple cloning site on the vector that we have, those restriction enzymes are not present on our insert DNA fragment on either side of the gene that we would like to express. So there is a method where we can artificially introduce restriction enzyme sequences in a PCR product by introducing them into a primer. So during the PCR reaction, we have two primers, a forward and a reverse primer. The forward and reverse primer bind to the target DNA fragment of interest in order to amplify that fragment. Primers are artificially created oligos that are manufactured in order to have sequence complementarity to the DNA fragment of interest. So if our restriction enzyme sequences in our vector are not present on the DNA fragment of interest, we can artificially introduce restriction enzyme sites into the primers. Restriction enzyme sequences are introduced on either side of the forward or reverse primers, including a spacer region. We also include a spacer DNA fragment in order to allow the restriction enzyme to bind to that DNA fragment. Because if we just had the restriction enzyme sequence itself, it may not have the correct conformation in order to be recognized by the restriction enzyme. So we artificially introduce a spacer and a restriction enzyme sequence on the forward and reverse primers. Now, during the PCR reaction, the forward primer will bind and amplify in the 5' to 3' direction, and the same with the reverse primer that will amplify the fragment. And during successive rounds of PCR, the spacer and restriction enzyme sequences are also amplified into the PCR product. So our final PCR product will then contain the insert DNA fragment with our restriction enzyme sequences on either side of that insert DNA fragment, as well as a spacer region that can allow for the restriction enzyme to bind to that fragment. Following the PCR reaction, the PCR product can then be exposed to the two restriction enzymes that we introduced the sequences for, and the vector can be restricted with the same restriction enzymes. and that will allow for the insert DNA fragment or the PCR product to be inserted into the vector in the correct orientation. And then we have a recombinant DNA fragment or vector that contains the gene of interest in the correct orientation, so this can now be used for expression studies. I'd now like to summarize once again what we've been discussing. We spoke about vector DNA sequences and insert DNA fragments. So this diagram shows two examples of the preparation of a recombinant DNA fragment. Either we can have a PCR amplified fragment of DNA, and that DNA fragment can be ligated to a vector. Alternatively, we can digest two vectors with the same restriction enzymes, and we can take an insert DNA fragment from vector A and ligate it to vector B so that it can be grown in a particular bacterial host. Once these DNA fragments have been assembled, we then have a recombinant DNA fragment. And this recombinant DNA fragment can then be transformed into E. coli. We'll now discuss transformation in some more detail. Transformation involves inserting the recombinant DNA fragment into a bacterial host. And when that recombinant DNA fragment is inserted into the bacterial host, that's now called a recombinant bacterium. There are two methods that are used for transformation. The first method that's most commonly used is heat shock. During the heat shock process, bacterial cells are first treated with calcium chloride. The calcium chloride makes bacterial cell walls permeable to DNA. DNA or recombinant DNA fragments are then co-incubated with the competent bacteria. After a time, the bacterial cells are heat shocked at 42 degrees Celsius for 30 seconds, and this heat shock forces the DNA to pulse into the bacterial cells. Once this is achieved, bacterial cells are then recovered for further use. The second method of transformation involves electroporation. Electroporation involves a brief but a high-intensity pulse of electricity that allows for recombinant DNA to enter the bacterial cell. Again, recombinant DNA fragments are co-incubated with bacteria. They're then exposed to an electric shock, which forces the recombinant DNA fragment into the bacterial host. Once the recombinant DNA fragments have entered the bacterial host, these bacteria are then recovered for further use. I would now like to summarize what we've discussed in this lecture. To begin with, plasmid DNA and the DNA fragment that we are interested in cloning are both cut with the same restriction enzyme or restriction enzymes. The restricted DNA fragments are then added to a linearized vector in the presence of a DNA ligase. When we talk about a linearized vector, we talk about a vector that has been cleaved with a restriction enzyme. So the insert DNA fragment and the linearized vector are ligated to each other to form a recombinant DNA molecule. The recombinant DNA fragment is then inserted into a bacterial host in a process called transformation. So transformation is not always a highly efficient process. And by this, I mean that in the process of transformation, vectors that contain insert DNA aren't always introduced into the bacterial host. In some cases, we could have religated vector or vector that does not contain the insert DNA, or we could have bacteria that do not contain any vector DNA or any additional DNA fragment. And so we need to select for the bacteria that contain our insert DNA fragment of interest. And in order to do that, there are various methods, and these are called selection methods. And these are the methods that we'll now go through in the next lecture. So that's it for this lecture on recombinant DNA technology. In the next lecture, we will look at the various selection methods that are used to select for transformed bacteria. Thank you.